Hi, this is Celeste from the Celestial Report, and boy, do we have a lot of new nanotechnology to talk about today. You're not going to hear it anywhere else, or you're going to hear it here first. So the first thing I'm going to do is, this little guy's toolbar is in the way. Let me get this out of the way. Eeny, meeny, miny, mo. Well, I don't want that one. What happened to my other one that was supposed to be up here? This is live, so didn't want to do that one first. My toolbar is just not big enough for all my, well, let me see if I can bring this one up and go on the side. Okay. This is the one that I actually want to start with. So this is my genetic engineering and biotech news, and it was from this morning. And so let's go through this. Researchers uncover a brain protein, a potential autism biomarker, and that immediately got my attention. So Northwest Medicine scientists sought to understand the link between children with autism and epilepsy, and they uncovered a critical brain protein that quiets overactive brain cells and is at abnormally low levels in children with autism. This protein can be detected in cerebrospinal fluid, making it a promising marker to diagnose autism and potentially treat epilepsy that accompanies the disorder. Now, there are several things. Here is the paper, and it was in the journal Neuron. And I've got to admit, there's some very provocative language going on here. I won't open up the study because then that will put you all to sleep. But neuronal ectodomain shedding. So these are neurons, and let's just, is my computer gonna, I might have to do this real time. I did have all my little tabs up here, and then the little tabs just like disappeared. So I want to look up ectodomain. And I wrote my caps on here. So ectodomain is this whole idea that surrounds shedding. And as you all know, we've been talking about shedding and transmission and all these different things. But in this particular, it's talking about the release of extracellular domains of membrane anchored proteins. Now, remember, right now we're in this process of genetic editing in direct evolution of the various membranes in our body, and they are anchoring these new proteins into us to direct our evolutionary process. And so I want a definition here. Let's put definition of ectodomain. See, my computer is just being terribly bad right now. Definition. Come on. DEFINITION. Definition of ectodomain. So these are parts of proteins. So they're protein fragments, and that's what we're getting in certain pharmaceutical products that come in contact with surfaces, which lead to signal transduction. And like I said, it has to do with shedding. But what is just the definition of ecto? Ecto means the outer or the outside or the external. So this would be outside of your cell. They're talking about it. The reason that I'm concerned is because there's this whole idea of ectoplasm, and I they first it let's just say it has some witchcraft type of definitions. A visible substance believed to emanate from a body of a spiritualistic medium during communication with the dead. And in today's webinar, we were talking about the elements, and is the heaven and earth already being burnt up as a result of scientific endeavors to break everything down to its elemental, basically energy, and then rebuild it and engineer it. It is a transparent corporeal presence, like a spirit or a ghost. So I wanted to bring this to your attention, but we've got some other. Let's see if I can get the right one. Okay, so and then we have another word that we've got, because this is a word that we have not heard about, the shedome. So let's bring this up, and I'm going to put define the shedome. So it has to do with the blood, and I did look at this before I even got online, and I couldn't find an exact definition for the shedome. Oh yeah, I forgot about that. So there is another word that we're going to have to learn. So basically the shedome, I am assuming from this particular, from ScienceDirect.com, talks about proteins that go from one cell transmembrane to another cell. And it could be through various influences, like we just mentioned in the ectoplasm. How much of it is chemical, how much of it is spiritual, I really don't know. But this is a word that you're going to want to pay attention to, the shedome, in the days to come, because it's going to become important for you. It's going to make news. Let's just put it that way. And another word that we have not seen come up on the map until now is the secretome. So I'm going to put that. And to me, this looks like a secret to me. I mean, they love to play little word games, but it's actually secretome. But it could be in the dual use language, secret to me or secret to my body. You're the only one that doesn't know about it. So it consists of proteins secreted from the cells. And once again, they're getting our cellular material, natural original cellular material out, and the edited material in. So these are some new words. There's also some other another thing that I'm concerned about with this is this protein is a biomarker, but what they like to do is put either hydrogel or quantum dots on the protein as a biomarker, so that with their various imaging tools, they can pick it up. And they're also going to be doing this with Parkinson's patients as well. So I wanted you to be aware of that if you do have an autistic child, be aware of this fluorescent biomarker protein, and it will be presented to you as a good thing that they can watch the shedome and secretome and basically bring homeostasis, which is the balancing of your neuronal network. But, you know, once again, here you've got, but you can shed the ectodomain, and I would say shed the ectoplasma domain, as in spirits, ghosts, and other types of things. But another thing that it ties in, and actually it was quite amazing that I found this like a week ago before this study even came up. So we'll X out of this one. I just want you to see that this is from, so that you can see that it's from Genetic Engineering and Biotechnology News. It was today's. Now I'm going to go over to the history of precision medicine. Precision medicine is a medicine that is based upon your blood. And basically your treatment is going to be determined by your genome, and they're going to treat you through your genome, and you're no longer going to have drugs or surgery. But I want you to see how far back this autism thing goes. It was, I mean, I was looking at the personalized medicine, but then the autism thing really caught my eye. So just so you know, the first blood transfusions were in 1907. Autism. So first blood transfusions, 1907. The first autism was used to describe a subset of schizophrenia. And one of the side effects of certain things that we're seeing right now is mental illness, especially schizophrenia, as it relates to autism. And that was back in 1908. And they do give you a link over here. This is a history of precision medicine. Now, going on down, we have recombination of the genes in 1911. That really surprised me, that there was recombination of chromosomes going on that early. The first genetic map was in 1913, according to Nature magazine. Epigenetics, that is your RNA expression, was 1942. Dr. Canner, MD, publishes a paper on the rare disorder of infantile autism. That was in 1943. And then we go on down. Then really heavy duty stuff with the genome goes on. In 1959, they began to have the ability to switch genes on and off. There it is right there. They were able to turn certain things on and off. And that's why some of you may be thin, some of you may be fat, some of you may have these unexplained medical conditions. The food in our country has certain chemicals in it that other countries don't have. So if you drink, let's say, a Coca-Cola, which I don't advocate in America, it is going to be a different formula than what you get in Mexico. The first supercomputer was in 1960. The genetic code was cracked in 1961. The first chatbot was in 1964. In 1967, the Autism Research Institute was founded. They already had, look at how much money they were bringing in, and they had the belief that autism can be cured. Well, of course, if you create the problem, then you can cure it. Pharmaceutical treatments for autism in 1970 become the main treatment method. Twin study, 1977, with autism. Almost sounds like Nazi 1980, the code, the medical code describes autism for the first time in 1980. In 1980, the autism incidence is 1 in 10,000. I don't even know how much it is now. In 1987, they expand the autism to a spectrum. If you're seeing a pattern here, well, there's other things going on with the genome. Behavior analysis therapy can treat autism. So they had this problem that wasn't a problem a long time ago. And then all of a sudden they've been playing with various things like behavior, this thing, medical treatment. That is called experimental evolution, where you play with different things to try and see what you can do with it, if you can direct the evolution or not. 1995, the Cure Autism Now was founded, Defeat Autism Now, 1995. See, out of all of the diseases, they chose autism, and it's really interlinked with this whole genetic program that is rolling out. Autism incidence in 1997 is 1 in 5,000. And I won't go into all of these. It goes on and on and on, you know, genome, autism. So they were really playing with these children. And it's very, very sad to see it mapped out here the way that it is. So I am going to bookmark this because I lost it just before this. And we're going to go on to another article in Genetic Engineering Biotech Today. And that is the SARS-CoV-2 is neutralized by antibody-like proteins from sharks. Now, most of the time when you see antibody-like, that means that it is a synthetic versus a natural protein. And these are called VNARs. You're going to need to watch out for that, especially if you eat kosher food, because this does come from a shark. I don't know if it makes a difference if it's a synthetic shark cell versus a natural cell or natural shark cell. I guess what I'm concerned about is there is the original construct of the SARS-CoV-2 had 7 different animals and 7 different pathogens associated in the construction of this construct. And now we are starting to see additional genetic material from different animals being added to this construct. I don't have the complete list, but I've been tracking it over the last several weeks. And now they're adding sharks to it, as well as other animals. But remember, it started out supposedly only as bats. And then supposedly it went from bats to humans. So I just want you aware, supposedly this can't reproduce, blah, blah, blah. But they're also applying it to the SARS COVID or the SARS virus, which is very similar, and the MERS, which is your camel version of this whole thing. So it's something I just wanted to bring to your attention. I don't know that there's much that we can do about this mixing of the species. This is called xenotransplantation. In the 1990s, every person that was xenotransplanted died. So I'm just letting you know that if therapeutics come out I don't know about, I don't have the statistics. I'm not a statistician to know, if you would die or not in this particular case. Let me now go to, where did that go? Just a sec, one more. Wasn't the sharks, wasn't the brain. Oh my goodness, where did my other story go to? Wonder if I can just go like this and I know it was here. I know I had it up here. Well, what I guess I will just have to wing it on my own. That's why I have a memory. Let me clear out some of this. It's got to be in this one. That's that one. And that one. I think they may have taken it down since. Very interesting. Oh, there it is. Okay. I'm not seeing things. Okay. So this is the last article that we'll look today, but it also has some really interesting. This is fridge-free vaccines. And remember that the ones that we have right now have to be at these super, super cold temperatures, which is a barrier to being able to distribute vaccines in either hot or cold places. And so this one company called Stable Pharma, and I'm going to take you to their homepage right now, has developed a novel and disruptive technology that aims to produce a fridge-free vaccine to eliminate the need for the global cold chain, to reduce vaccine wastage and save lives through better immunization. And then they have this longer piece, but I am going to have John play the other piece, and then we will talk about possibly the problem with this particular solution that they have. So, John?
Okay, and if you could unshare your screen, Celeste, that will make it more viewable for peeps.
Okie doke.
The secret to this invention lives in the deserts of the world. It's a plant called the resurrection plant, and it's developed an amazing ability to survive years of drought by using a special sugar in its cells called trehalose, which effectively puts the plant into a state of suspended animation. When the rains return, it absorbs the water and switches back on, coming back to life again. StableVax uses the same sugar from the resurrection plant, but in a tiny sponge inside a standard medical syringe. The sponge contains the trehalose sugar, which stabilizes the vaccine, making it completely inert. When the vaccine is used, the syringe is filled with sterile water. The small sponge inside now releases the active vaccine, and in seconds it's ready to inject into a patient. The secret to this
Okay, so now what could be the problem with this? So if you went to my pathos on our Great Delusion, you would have heard between that and my Dark Winter presentation that they were planning to use the resurrection plant and the tardigrades, which you all know are the little water bears. And why? Because they wanted to start in directed evolution to get humanity to a place where they would not require water, they would not require air, that they could live in extreme temperatures. So they, through directed evolution, they edit the human body and mix human genes with the little water bears and the resurrection plant. I did get this from military and other, military and Homeland Security documents. There are webinars. You can look it up. You can look at my coverage if you have not heard this, if it's startling to you. One thing that I did not know is that if they use the resurrection plant formula, that the resurrection plant actually promotes 2 different strains of C. diff. And C. diff is an infection that you get from being on antibiotics. It causes severe gastrointestinal problems and even death. And so they are in the process of adding what you just saw to all of the current vaccines. So they are rewriting the vaccines that are out there, and they will have a different formulation, a different formula, including the resurrection plant. You could read all about it at this, at this company, Stable Pharma. You can read all about their project to get more details. I do believe the tardigrades are already in the current, so that would not require a change of the ingredients or the recipe, if you will. So I guess that's all I have for Nano Moments. That's enough for today. But I think these were really important things to get on your radar, some new words, some new concepts, latest in gene editing and what's going on. And I will be back at the journals after the holidays. But if something does pop up, it seems like there's a steady stream of new technology that's being deployed right now. So I will keep you posted even through the holidays, even if it's just something brief. So thank you for your interest in Nano Moments, and we'll see you the next time.